Welcome everybody to the Ernst Abbe Award session put on by the New York Microscopical Society. And I'll pass this over to my co-chair, John Refner.
The Ernst Abbe Award was established back in 1973. Brian Ford is our recipient this year.
Our speaker is our award winner, Professor Brian Ford. He's an active author and lecturer in a wide variety of scientific disciplines, and he often uses microscopy to solve a variety of problems, not only scientific but just general world problems. And he has authored over 35 books. And his latest book, Non-Science Returns, is released. We'd like to thank Dr. Ford for being our awardee and attending the symposium today. And here's Brian.
It was my vintage Lights microscope that first opened a magical door to me. Unlike Narnia, my realm was real and rich in remarkable revelations. Optical instruments fascinated me from when I was very young. They could tell us so much about the world that was beyond our sight. And it was microscopic revelation that was to preoccupy me through the whole of my scientific life. I have, from my mid-teens, some watercolour pictures of the cultures and the organisms I could see under my microscope, and this is the first microscope I had. It was little more than a toy, a little German microscope with concentric lenses. But it still allowed me to take pictures of bacteria and of pond life. And when I had my Lights microscope, shortly after my 16th birthday, I used it to build Fred, photomicrographic recording and enlarging device. It gave me some decent pictures of plant histology. And look at those beautiful ribbon chromosomes from a species of bluebell. Microscopy as a hobby allows you to work throughout the night if you wish, and I was always captivated by the revelations that the microscope could bring. The first organism I ever tried to film down the microscope was Hydra, in a drop of pond water. Like so many organisms you find in a pond, it has a ballitic beauty. No child should get to the age of 10 without having seen pond life through a light microscope. It alters the way you look at the whole of life. Over the years I accumulated thousands of permanent preparations, but I always greatly enjoyed the engineering side of it all, and I adapted a camera from a World War II fighter. Here is Spirostoma, one of my favourite ciliates. And under high magnification, with the substage a little bit off centre and the light cut down just a little bit too much, you could begin to discern some of the miraculous structures that lay within the cell. I was intrigued also by what you could see in living blood. These stills are from a film I made at the time, showing how you would prick the finger, pick up a blood drop, dilute it with physiological saline, and then add a cover slip. You could see red cells iridescent against the black background, and white cells and cells from bone marrow. How much this could reveal of the majesty of life. One day a friend called, he was an angler, and he said that fish in the nearby lake had been acting strangely, sluggishly. They seemed to be unwell. Under the microscope, using my live blood technique, I could see tiny flickering organisms, and I drew one. It was, of course, a trypanosome. I managed to take pictures in black and white, and then on 35mm Kodachrome. Sometimes The red cells seem to be stuck to the trypanosome, and I often wonder whether this is a hitherto unrecognised feeding mechanism for these protozoa. These studies were eventually published in the Microscope, and over the years I published quite a number of different papers on aspects of blood. Culminating in a single drop technique, it was quite simple. You take a simple drop of blood, allow it to coagulate on the slide for perhaps 7 or 8 minutes, and then judiciously add a cover slip, and the centripetal forces cause the formation of pseudocapillaries within which you could observe flowing blood as it coagulated. The extraordinary deformability of erythrocytes became apparent. Once in a while you'd see a cell that was stationary against the flow, as though held like a balloon on a string. Sometimes cells had a string coming from each end and were pulled out like an elongated cigar. Sometimes the filament would break, and on other occasions you could observe the filament, but as you increased the rate of flow by pressure on the cover slip, it would disappear beyond the limits of resolution. I took still photographs. You can see the moving erythrocytes, which present as blurred images, and these captive cells, pendrocytes I call them. And eventually, by dark field, I was able to capture pictures of the threads themselves. They are beyond the limits of conventional resolution. It was an extraordinary revelation, and led to my first ever lecture at the age of 25 in the rooms of the Royal Society in the centre of London. It also gave me my very first chance to speak to the Intermicro Conference in 1969, and I've done so almost every year since. It ended up featuring in McGraw-Hill's Yearbook of Science and Technology, facing a page of photographs sent back from the moon. Can you imagine how excited I was by that? On television I would sometimes show some of my films, including studies I made of head lice. People never talked about head lice. They were almost ashamed to admit they existed, but head lice were quite commonly found amongst the hair of Cardiff kids, and to me these were beautiful, translucent little creatures, even though everybody else in the television studio was scratching their head like mad. The still pictures turned up in medical magazines and in encyclopedias. Also in the newspapers was this terrible tragedy at Aberfan in South Wales, when a coal tip collapsed and nobody could find out why.
What makes a tip slide? The conventional theory is that when too much water is present, it lubricates the ground beneath the tip. If this is clay, it becomes as slippery as a banana skin, and suddenly the whole lot can shoot down the hillside. A Cardiff scientist, Prof Ford, holds an opposite view. He believes the cause lies in the tip itself.
The principle of it is quite simple. In a coal tip, the material runs up a kind of railway on a track of some description. Usually, of course, this railway runs on the surface of the tip. And the material discharges into a large conical pile. The angle of this is critical and is known, here it is, for obvious reasons, as the angle of repose. It depends on the way in which the different particles within the mass are pressing on the particles beneath them. And this depends on their density. When a mass becomes filled with water, saturated with liquid, the density of each particle is less. Therefore they press less heavily upon each other. The effect of this is to lessen the angle of repose. The only way this can happen is by the whole tip losing height, spreading out at its base, and in effect, sliding down onto the surrounding countryside. With this device, one can introduce water into a dry imitation of the slag that is found in South Wales coal tips. As the water soaks the pile, it becomes dark and begins to glisten, and sure enough, at a certain critical stage, the whole thing slides away. A rather terrifying example in miniature of what happens when a tip collapses.
It was also the microscope that solved the mystery of the Landok skeletons. I was asked to come in by the South Wales archaeologists and took a 16mm film of my investigations. I was actually the first private citizen ever to be given an exhumation licence by the Home Secretary. The skeletons had been found in a shallow grave on the outskirts of the capital city of Wales, Cardiff. Back at my little rented house that I had then, and that's my first car, it cost $40, I set to work. I discovered the brain had been deformed. A plaster cast showed a cherry-like growth on the upper surface. The murder weapon also emerged. A stone spear, a stone spike, had been driven through the centre of the head. So we recreated Joanna's skeleton, the first time for perhaps 800 or 900 years. But again, it was the microscope that gave the ultimate revelation. Some of the body seemed to have been butchered, and there were butchering marks on some of the ribs. One of them in particular showed small, dark fragments. And when I teased one out and analysed it, it turned out to be iron. Yes, so we had evidence of butchery, we had the murder weapon, we had the site of the fatal wound, and if that cherry-like growth had caused this woman to act in a strange way, we might even have a motive. She could have been killed as a witch. It was on TV, it made headlines in the newspapers, and also became the subject of a standard book on skeletons published by UNESCO. I also experimented in my home laboratory with taking film of feeding protozoa on standard 8mm colour film. This, of course, is Stencil. These organisms are free-swimming Vorticella, normally anchored down on their spiral stalk, but sometimes they swim free. And watch this ciliate grazing on the surface film of bacteria, leaving a clear path in its wake as it goes. In the late 70s I did my first ever world lecture tour, and please notice inside the plane, no overhead lockers. You used to put your coat on a shelf in those days. I had great times travelling on the trams, the famous trams in Melbourne. And of course I appeared on television to explain how microbes purify the water supply. With my pilot friend Richard, I flew up along the Gold Coast into North Queensland and went to explore inside the rainforest. I suddenly then realised that there was a possible mechanism of explaining why plants drop their leaves, even in the luxuriant surroundings of a rainforest. The plants were shedding their leaves. We've always been told that those spectacular autumnal colours come about because of chlorophyll breaking down, but microscopic revelations told a different tale. The palisade cells of an acer leaf in section become slowly populated by colours, and also by other waste materials. So I came up with a new theory published in Nature on what was happening when the leaves turned red.
Professor Prof Ford believes it's a tree's way of getting rid of waste matter.
They're not dying, they're doing something. If a tree contains, heavy metals and other toxins, they get pumped into the leaf before the leaf is fallen. So my theory is very simple. When the plant drops its leaves, that is the plant's mechanism of excretion.
He even believes it could have a practical application.
We do know that some trees will grow in heavily polluted soils. So suppose you had an area of ground that was heavily polluted with, let's say, a heavy metal. You could grow the tree, the heavy metal would be taken up by the tree, deposited in the leaves when the leaves fall, then you could collect all the leaves and get the metal back.
I also brought microscopic revelations into a whole range of publication. In 1976 I published Towards a New Microscopy in the Microscope, with a number of leading articles in Nature looking at cancer research, at biohazard legislation when I first proposed that there should be the safe handling of viruses, and reminded everyone of the microscopic blind spots that still surround us. And what of the early microscopes? The picture on the left shows a normal living cell photographed under a single lens microscope, but if the lighting is adjusted properly you get the picture on the right. And I published a number of papers on what these early microscopes really could reveal. This is all Robert Hooke could see through his compound microscope, a blurry, chromatic image, but under a single lens microscope, this is what you could see. It was only the single lens microscope that allowed us to see the finest details of these living organisms. This is one made by Banks, used by Charles Darwin, by Hooker, and by Robert Brown, who gave us Brownian motion and first named the nucleus. These were beautiful little microscopes, fitted with a range of lenses, the smallest lens giving the highest magnification. These are orchid epidermal cells shown in a BBC programme hosted by Adam Rutherford, using the same lens, the same microscope and the same tissue. This is what I could see. When these microscopes were used properly, they could reveal so much more than anybody suspected. And these revelations appeared in a number of journals. Here's a 2-page spread from the work in Scientific American. Here it is in La Recherche, the French scientific magazine. In 1981, Sir Andrew Huxley invited me to go and look at Leeuwenhoek's original letters. And to my immense surprise, there were packets of his original specimens, lying amongst his letters and untouched for more than 300 years. I flew out to the Netherlands to use his best microscope and took this extraordinary picture of my own blood. And look in the top right-hand corner, there is a white cell, and you can quite clearly make out its nucleus. That's an extraordinary achievement for such a tiny microscope. And of course, hundreds of papers appeared. This is published in New Scientist, in Nature, in the British Medical Journal, Scientific American, Science Digest. It was published all over the place. There are over 400 publications in the bibliography from this period. And of course, the work gave rise to 2 books, both of which I'm sure you know, and which in their own way became classics. It was also on Leeuwenhoek that I gave my first ever evening lecture to Intermicro in 1984, and that soon became an annual event.
Yes, I was supposed to have a day to recover from jet lag. I was originally scheduled, I beg your pardon, to be on tomorrow night, but I was slid forward because somebody is going to be giving away ice cream tomorrow, all of which shows to my mind what a splendid sense of value for precaution.
And they were always booked to autograph.
I'm wearing the same glasses in that photograph as now. They were in fashion, they were out of fashion, then they come back in again. That's right.
Brian,
Put them in the drawer for a while.
Oh, thank you. That
This is how the muscle cells of a dinosaur appear under the microscope. No, they're not fossilized. This is a section of modern crocodile tail, but all reptilian striated muscle looks like this. I'd long believed that dinosaurs evolved in an aquatic habitat, but paleontologists insisted that they were terrestrial and they held their tails aloft. If so, those poor muscle cells would have to burn half the day's energy. Artists, like the very talented Raoul Martin, show colossal creatures poised on a muddy shore, whereas we all know that it couldn't even support a 5-ton elephant. My aquatic dinosaur theory gave rise to the longest book I've ever written, all triggered by revelations of those struggling muscle cells in the tail. It was also the microscope that revealed to me how human society had begun. We could sometimes find butchery marks on bones from tens of thousands of years ago, but how had mankind originally evolved between being an angry ape that could catch prey to being a hairless, weak and feeble creature? There must have been a mechanism to bridge the gap. And suddenly I realised what had happened. Humans must have been kleptoparasites, driving off wolves and other predators and taking meat from prey they brought down. That would explain how wolves and mankind first came to cooperate. The theory was launched in Laboratory News and attracted a lot of interest. But it was in the living cell that I always found the most exciting revelations. Do a Google search for a living cell and this is all you find. CGI images. Hardly a cell in sight. In 1976 I published Micropower, in which I tried to set the record straight. It was translated into Japanese. On a visit to Tokyo, I was actually taken out to see a recycling plant where some of my ideas on microbial recycling were being put into practice. This simple paramecium, this beautiful euprotes, Noctiluca, the organism that glows in the tropical seas at night, and in these wonderful sequences taken by Jeremy Pickett Heaps in Australia, we can see the edge of the cell exploring its environment. Cells were not little diodes. Cells were performing extraordinary tasks. And this is perhaps the most remarkable sequence of all. A filament of antithamnion has been crushed with a steel needle so that one of the cells is destroyed. And the cells at either side recognize the damage and seek out a way to restore the cell to life. This is nothing they evolved to do. In nature, if a dinosaur trod on an antithamnion colony, or a boulder fell down and snapped it in half, The ends would be a great distance apart. The only place in which a damaged cell can remain with the two halves in apposition is on the microscopist's slide. Yet these cells work out what to do. This is single-cell intelligence. It proves that our conventional understanding of life is wrong. We accept that everything in the body is controlled by the brain. And we accept that neurons act like transistors. If you culture neurons, you can pick up the electrical signals that they emit. Rather irritating buzzing sound. These are neural spikes. But stretch out the spikes on the x-axis and you can begin to hear what is going on inside each of those little sounds. The result is a haunting sound like seabirds on a cliff. We can actually hear the language of the mind. All of this posed the ultimate question, one put to me by James Naughtie on a BBC programme.
Can a single cell think? The biologist, Professor Brian Ford, is giving a lecture at Churchill College, Cambridge today. He is on the line to, I hope, explain. Professor Ford, good morning.
Morning to you, Jim.
We're talking here about...
During the interview, I played the recording of the neuron spikes and contrasted the traditional way of looking at life with my new revelations.
Well, we always tend to teach these days that thought and data processing in the brain goes on at the synapses, the junctions between brain cells. I don't believe that. I believe that thought is going on within the nerve cells. I don't see us, you see, as bodies divided up into cells. I see us as great communities of independent, almost free-thinking cells that come together to produce the effect that we know of as human life.
Well,
Jim, you've gone suddenly almost speechless, which I have to say is uncharacteristic for you.
Well, no, I mean, just, after a description of what is the nature of life, it's a question of where you go next, really. Professor Brian Ford, we know you're lecturing at Cambridge today. That really was extraordinarily interesting conversation. Thanks very much indeed.
The interviewer was reduced to a state of shock. Nobody ever thinks of microscopic revelations. When Disney made Honey, I Shrunk the Kids, the floorboards just looked like the floorboards looked as they do to the naked eye, but larger. I processed the picture to show what you would actually see if you were that small. The same with A Bug's Life, where all the leaves look like sheets of green plastic, no cells to be seen. I added cells to this picture. Those, by the way, were engraved by Robert Hooke in April 1663. That's what a tiny ant would actually be seeing. So if cells are autonomous, technically speaking, they can live on their own. The body doesn't need the head, and the head doesn't really need a body.
Let's all welcome our evening with Brian spokesperson, Brian J. Ford.
Well, thank you very much. This is a gory and ghoulish talk, and I apologise in advance of the fact that it follows dinner.
The artificial blood circulation ensures the metabolism necessary for the life of the head. The isolated head lives on for hours, even reacts to light and to sound.
So if the head can exist without the body, can the body exist without the head? Mick, a chicken who was decapitated in 1945, lived for 18 months, being fed food with an eyedropper. One of my greatest pleasures a few years ago was to be presented with two previously unknown Lavender microscopes in the space of a year. This is the most interesting one. It's made of brass. That's what it looks like, and for the very first time I put it under the scanning electron microscope so that we could see the fine details of just how it was manufactured. This one was owned by a Spanish collector, and the mayor in Spain held a reception to celebrate its announcement. Leeuwenhoek seems to have destroyed the lens before he threw this one away. The edge of the lens shows in blue, and I was able to restore the rest of it so that we could calculate its magnification. In fact, when the Royal Society very kindly elected me to honorary fellowship recently, they used one of my diagrams to headline their announcement. And what of yeast? This is how Leeuwenhoek drew it. That's what it looks like under a single microscope. And here are a couple of pages from Encyclopaedia Britannica in which I explain how these microscopes were used. It is fermentation that produces the taste of chocolate in the cocoa bean. It is fermentation by single cells that creates the flavour of our best salami. And what about single cells as a form of food? French television came across to ask me about my views on how single cells in future could feed a growing population. I explained that the way that we farm meat is not sustainable in the long term, and single cells could provide an answer.
Le biologiste Brian Ford n'a pas attendu le premier hamburger in vitro.
But only if we sweep away the old ideas and allow ourselves to embrace new ones.
C'est trouver un moyen de produire plus de viande pour satisfaire la demande croissante, mais sans solliciter encore plus l'environnement, et la viande de synthèse est une des possibilités.
Spontaneous human combustion was an unsolved mystery, and I realized living cells could reveal an answer. The cells of people, when they aren't very well, can generate acetone, and acetone produces an explosive vapor.
Professor Brian J. Ford is a research biologist and an author who's lectured at universities including Cambridge and Leicester. He's also published work on the subject of spontaneous human combustion.
It's a very nice, quaint little neighbourhood.
It is nice. What's the mystery surrounding spontaneous human combustion?
The point is that human bodies are profoundly non-inflammable. That's the mystery, and that's what I wanted to solve.
The recent research being done in Cambridge, England, might offer a whole new insight into the phenomenon. Professor Brian Ford is a leading independent researcher in cell biology. He believes he's identified a substance that could solve the puzzle. Something so volatile, it can ignite with just an invisible spark, like the static from a sweater.
It will set fire to the bottle and it can almost explode in your face. I set out to search for a compound in the body that could burst into flame. And although we often overlook the fact, human bodies produce acetone.
My scale models proved the point. In 1986, BSE, mad cow disease, appeared in British cattle. Cattle would tragically collapse, their brain destroyed by abnormal proteins, prions. And by 1996, we realized it had spread to the human population. I discussed it on TV and with Carlton Gadjuchek, American scientist, the man who discovered the prion and was also jailed for molesting teenagers. From this sprang my book, BSE The Facts, a book commissioned, researched, written, printed and bound within 28 days. I think that's a record for scientific publishing. Cells also gave me the revelation that plants have memory. When dandelions are repeatedly mown, they learn how to respond. The light microscope and the SEMs at the Cavendish Laboratory in Cambridge allowed me to show the changes in cells that allow the plants to hug the ground. There was also the revelation of bacteria as particles of nucleation that cause rainfall. That revealed to me how the rain dance could work. If your feet are pounding and driving up dust from the ground, you would allow bacteria to ascend into the heavens, and that could indeed have caused rain. Perhaps the rain dance was real after all. I was asked to write a manual of the first of the digital microscopes. One of these was given to every school in Britain. It allowed me to produce a fully illustrated manual. And indeed it was translated into Welsh, and became the first such book ever to appear, translated into that ancient language. My little granddaughter Lucy, now at university, proved just how easy it was to use. My wife and I used to travel on the QE2 as passengers. Little did I think that 20 years ago I would become one of their guest speakers. The important thing about lecturing is that you must keep to time. Don't start late, or you have to catch up.
Good morning, everybody, and I'm very sorry to start a little late, so I've only been fast otherwise, because otherwise I'm not going to get through the entire thing by the time I finish what I could really do. I mean, I had several clicking with all the stones and various things and so on. So I shall get the lecture exactly what it's going to be, but of course I have to see that just as good, and I hope very much that you don't lose any of the actual sense of it, because it's very important that you try to get the general nuance, even if the occasional word falls by one side, which of course inevitably happens whenever you happen to start a little bit sooner.
Thank you for sharing with me some of the revelations that the marvellous microscope has given us. From the complexity of the living cell and the hidden beauty of life, revelations of how blood coagulates, how head lice behave, how coal tips collapse, solving an ancient murder, seeing how plants excrete and yet have memory, from giant dinosaurs to tiny intelligent cells, secrets of the brain and the language of the mind, cells in our food, cells making food, cells becoming our food, and even showing how it could be that people tragically burst into flame. Even the rain dance may have an answer through microscopical revelations. Microscopists love new ideas, so let me leave you with my closing words from the plenary lecture I gave to the Microscopy Society of America. If there's one thing microscopists like, it's new answers. Not answers to conventional questions, but answers to questions that have never before been asked.
This is a great and glorious discipline in which we all live. And I want to leave you with this picture. This is what Leeuwenhoek saw. He was looking through beautifully made lenses at fabulously detailed images and was laying the groundwork for the discipline in which we all find so much satisfaction today. What we've been told is filled with errors, and it's high time we woke up to it, because this image is the origins of microscopy and in many ways the roots from which today's bioscience era has sprung. Ladies and gentlemen, thank you very much. Thank you. Excellent. Thank you very, very much. Thank you all. Thank you.
Thank you so much, Brian, for an amazing talk. And it's really remarkable, all of the answers that the microscope has been able to provide. And John is going to present the award.
I want to pass the medal on to you, okay? Everybody has seen the medal up here. Yeah, there's the medal.
And as if by magic, it ends up digitally in my hand. Thank you so very much. What an extraordinary honour that is. I'm so very grateful. Marvellous.
It's a great honour to have this, and I'm delighted and honoured by your joint attendance this afternoon for you. So thanks a lot, everybody, and goodbye from us.